Hello once again, my children. Let's just recap what we have done in genetics thus far. We looked at the terms, we looked at complete dominance, incomplete dominance, codominance, sex-linked inheritance, blood groups, pedigree diagrams, and now dihybrid cross. Of all the crosses, children find this a bit challenging. That's because they don't break it down. And if you break it down as is required of you, you should have no problem. So let's go through it and let's see if you understand. That's why we've been looking at monohybrid crosses. Monohybrid cross is a cross involving one characteristic. For example, the color of the flower, one characteristic. And the shape of eyes, for example, almond shape or slit shaped. Texture of the seed, smooth or wrinkled. That's one characteristic. Now, let's recap. One characteristic controlled by one gene, and the one gene has got two alleles. I repeat, one characteristic controlled by one gene, and the one gene has got two alleles. Those two alleles you represent using two letters. So thus, if you have two characteristics, you must know that when two organisms come together, multiple characteristics come together from both the parents. We're just doing it so simply. So if there are two characteristics, there'd be two genes controlling it. If there are two genes, how many alleles? Let's go back. For one gene, two alleles. For two genes, four alleles, 2 times 2. So whilst we used two letters to represent an organism in a monohybrid cross, how many letters will be used to show the genotype in a dihybrid cross? 4 letters. Are you with me, darling? So, for example, if I said a tall plant crossed a short plant, so tall and short refer to height, one characteristic. So I can represent the tall plant as capital T, or capital T, small t, only 2 letters. Short plant, I will only represent as small t, 2 letters. Now, if I'm doing a dihybrid cross and I say a tall plant with red flowers, and I say red is dominant, how will I show a tall plant with red flowers? Let's look at this, my darlings, okay? Let's look at this. So, sorry. So, tall. 2 ways I can show tall. Capital T, capital T. What is the other way in which I can show tall? Yeah? Capital T, small t. So those are 2 ways. Are you with me? Tallness as a characteristic, one characteristic. Okay. So I say a tall plant with red flowers. Okay. So if I want to show red and I say red is dominant over white, I can either show red like that, or I can show red like this. How's that? So let's put them together. So what will the genotype of a plant be if it's tall and red? What are the possibilities? What are the possibilities, if a plant is tall and red? Let's look at the possibilities. Tall. Which are the various ways in which I can write tall? So let's write it that way. Red. So that's tall and red plant. Is there another way? Is there another way, my darlings? Tall and red? Oh, I could write. Would I also be right? That's a tall and red plant. Is there another way? Yes. Tall and red. How's this? Capital T, small t. Is that tall and red? Yes. Okay. Let's see another way. Another way in which I can write tall and red. You look at it and tell me. So that's one way in which I can write tall in red. There's another way, tall in red, because tallness is dominant and redness is dominant. Here's another way, tall in red, as long as I got a capital T and a capital R somewhere. How about this one? Tall in red. So there are 4 ways in which I can write tall in red. 4 ways. Are you with me, my darling? It's like saying that with that, that with that. Can you see that there? That with that, and that with that. That's exactly what I did there. I took this and I combined it with that. I took that and combined it with that. I took that and combined with that. And those are the 4 possible ways of being tall and red. So in this particular question about dihybrid crosses, you need to find out the various genotypes organisms can be. So that's how I can represent tall and red. So tallness was dominant, shortness was recessive. Redness was dominant, whiteness was recessive. So let's take this out and you tell me how I could write tall and white. Okay, tall and white. Let's see how I could write tall and white. Okay, so tall, capital T. White is a recessive characteristic. Small r, small r. Is there any other way in which I could write it? Is there any other way in which I could write it, my darlings? Yes. Capital T, small t, small r, small r. So there's only one way in which I could have written short, small r, small r. And there are 2 ways in which I can write tall, capital T, capital T, capital T, small t. So you could be tall and short, tall and white. So the first one I looked at was 2 dominant characteristics, tall and red. Now I'm looking at 1 dominant, 1 recessive, tall and short. Let's look at, tall and white. Let's look at short and red. So once again, 1 recessive, 1 dominant. Short and red, okay? Let's look at short and red. Short and red. Now, short is a recessive characteristic, isn't it so? And a recessive characteristic can only be written in 1 way, with 2 recessives. So, short is recessive, so small t, small t. Red is a dominant characteristic. You can either be homozygous dominant, or you can be this. Short is only 1 way. There we go. So you can be that genotype or that genotype if you're short and red. Let's recall. Tallness is dominant, redness is dominant. So you can be tall and red. Or you can be red and short. Red and short, like this is red and short. Or you can be tall and white. White is recessive, tall is dominant. What is the last combination? It could be. What's the last combination? 2 dominants, 1 dominant, 1 recessive, the other dominant, 1 recessive. The last one would be 2 recessives. Short is recessive and white is recessive. Recessive, there's only 1 way to write it. Okay, my dear children, that's it. So there we go. Those are the various genotypes. How many letters did I use to show the genotypes? 4 letters, because for 1 characteristic is 2 alleles, 2 letters. For 2 characteristics are 4 alleles, and therefore 4 letters. So this is the genotype of a plant. So the genotype you will write using 4 letters. Okay, the gametes. What would the gametes look like? So let's look at what is written over here before we proceed. A dihybrid cross involves the inheritance of 2 characteristics. Mendel explained the results obtained from dihybrid crosses according to his law of independent assortment. Now, what on earth is independent assortment? According to this law, alleles of a gene for one characteristic segregate independently of the alleles of a gene for another characteristic. So that means these two alleles separate independently of that. So in other words, if I had to form a gamete from here, the gamete will have one of these two, for example, a small t, and the gamete will have one of these two and a small r. So in this case, all your gametes would have small t, small r. So learn this off by heart, where the alleles of a gene for one characteristic segregate independently of the alleles of a gene for another characteristic, and the alleles for the two genes will therefore come together randomly during gamete formation. So let's take another genotype, my darling. Let's take another genotype. So if I take a genotype like this, tall and red. Red is dominant as well. But I've got this here. So that means the capital T, one of the capital T's will go out, for example, if there's a male, into a sperm, and one of these here will go out into sperm. So you can get a sperm with capital T, capital R, and you can get a sperm with capital T, small r. So they separate out independently of each other, and then they come together in the gamete. So in this case, every gamete will have two letters. In our monohybrid cross, where we had a genotype, for example, capital T, small t, representing a tall organism, and when the gametes are formed, that capital T and small t, by the law of segregation, separate out such that each gamete has only one letter. So you'll have a gamete with capital T, another gamete with small t. But now you've got two characteristics. So each gamete will have two letters, one allele from each characteristic. And to work out these gametes, I always suggest to children, use a Punnett square to work out the gametes. And now I'm going to show you how to work out the gametes using a Punnett square. So let me just erase this, my darlings. Okay, so if we have that, okay. Sorry, my darlings. So if I have capital T, small t, capital R, small r, right? If this was my parent, okay, let me make this bigger. If this was the genotype of the parent, what would the gametes look like? So the gametes will go out in these combinations to their, sorry, the alleles will go out in these combinations to the gametes. You can have one gamete with capital T, capital R. You can have another gamete with capital T, small r. It's like mathematics. You can have another gamete with small t, capital R, and another gamete with small t, small r. This gives you the greatest variety of gametes because both characteristics are heterozygous. Okay, so if you want to work out that gametes, my darlings, if you want to work out the gametes, what you can do is to just, in a Punnett square, put a capital T there, and then a small t there, okay. Okay, and the other alleles for the other characteristic got capital R there, and the other one, what is the other allele, my darlings, up there? One is capital R, the other is small r. So those are the alleles. Now I'm going to see now if that gamete met that one, if that gamete met that one, what would my first possible offspring look like? If that one met that one? Sorry. if that one came together with that one in the gametes. So these are now the gametes, my darlings. These are the gametes, not offspring. Okay, so you get that. And if that allele met that allele in the gamete, remember I told you each gamete will have 2 letters as compared to the monohybrid cross where each gamete had 1 letter. So there are 2 characteristics, you'll have 2 letters. Okay, so what would happen if that allele met up with that allele in the gamete? You'll have small t, capital R, there. And here, my darlings, we would have that and that coming together, small t, small r. So these are the 4, let's just show it to you like this here. So I can draw the spotlight and show you. These are the 4 possible gametes. These are the 4 possible gametes. Now we're going to see what's going to happen if these gametes met each other. If these gametes, so if these are the 4 possible sperms, if they meet 4 eggs with these types of allele combination, what could the offspring look like? Okay, so we're going to look at that particular cross in the next lesson. But suffice for you to know for this lesson, oh, there's a mouthful there was this time, that in a dihybrid cross is a cross involving 2 characteristics. One characteristic is controlled by 1 gene, and 1 gene is represented with 2 letters because there are 2 alleles. So if there are 2 characteristics controlled by 2 genes, each gene has got 2 alleles, so there'll be 4 alleles you represent by 4 letters. So these 4 letters, for example, will be the genotype of a plant, okay, for a dihybrid cross. And we said that during gamete formation, these alleles for each characteristic separate out independently of the other and then come together. So this one here will separate from that. So this will be found in one gamete, and this will be found in the same gamete. That will be found in one gamete, and that will be found in the same gamete. So they come together randomly. That's the law of independent assortment. And then what will happen is that this gamete may meet that one, but it can't be, if these are all sperms. Then if you have eggs of the same type, this may meet a capital T, capital R of an egg, and then you'll get out a new genotype. That's what we're going to be looking at in the next lesson. So my darlings, go and read through this and read through this in advance before we look at the next part of dihybrid process. God bless until I see you again.